Welcome to this lecture series on eukaryotic gene expression, basics and benefits. Today we are going to discuss about a very fascinating and very interesting area of regulation of gene expression in eukaryotes, that is about regulation of eukaryotic gene expression by small RNAs, a phenomenon known as RNA interference or RNAi. Now, the last few lectures, especially from lecture 29 to 36, we focused our attention primarily on expressing genes. Like we started with basic cloning and expression vectors, and then we looked at some of the basic expression systems in prokaryotes, and then we moved to expressing genes into yeast and insect cells. Then we discussed about viral and non-viral gene delivery techniques for expressing genes in mammalian cells. Then we looked at some of the applications of expressing genes in vivo. We discussed extensively about human gene therapy, how by expressing genes we can cure human diseases, or at least we are attempting to cure human diseases. We still have not succeeded. Then we moved on to another very interesting area called as DNA vaccines, how expressing antigen genes can lead to evoking an immune response leading to DNA vaccination. Then we discussed about transgenic animals, transgenic plants, and in the last class, instead of discussing about expressing various genes in various systems, we discussed about knocking out genes. So, we discussed about knockout mice, knockout rats and so on and so forth, where technology was developed to specifically knock down or knock out the expression of a particular gene, so that the protein cannot be made, and how this knockout technology has been very useful in understanding gene function and why this technology of developing knockout ultimately got the Nobel Prize. Today we are going to discuss about another very interesting area of again repressing the expression of genes in eukaryotes, namely regulation of eukaryotic gene expression by small RNAs, and I am going to discuss about or introduce to you a very fascinating area of research known as RNA interference or RNAi. What is RNA interference? Let us first define RNA interference and then try to understand what RNA interference is. RNA interference or RNAi, as it is popularly known, is an evolutionarily highly conserved process of post transcriptional gene silencing. Remember, this is important. It is a highly conserved process of post transcriptional gene silencing, that is, this silencing happens after transcription, by which double stranded RNA causes sequence specific degradation of mRNA sequences. Remember, RNA interference is a post transcriptional event. It involves formation of double stranded RNA, and once double stranded RNA is formed inside eukaryotic cells, that somehow silences the homologous gene sequences. So, the expression of the corresponding genes which has homology to the double stranded RNA gets abolished and leading to loss of expression of that particular gene. You will be able to understand better once we explain what is RNAi, then the definition will become much more clearer. As I said in the last class, I had discussed about knockout mice, that is the technology for developing the technology for knocking out genes specifically in the germline, leading to creation of what are called knockout mice was one of the important landmarks in the area of eukaryotic gene expression, and because of its importance, the people who discovered this knockout technology were actually awarded Nobel Prize in the year 2007. Now, today again we are going to discuss about another Nobel Prize winning work. The Nobel Prize for discovering RNA interference was awarded in the year 2006 for Andrew Fire and Craig Mello. So, RNA interference is a very important discovery, and since it has been awarded Nobel Prize, that means it has really had profound implications in the area of biology and medicine. So, let us now try to understand what is this Nobel Prize winning work of Andrew Fire and Craig Mello, and how exactly RNA interference has changed research in biology and medicine. So, as is important, it is always important to understand the history of some of these important developments that took place in the area of biology and medicine. So, I am going to briefly tell you about what is the history of RNAi, how is this RNA was discovered. Now, at least we can trace back to the year 1990, when the first concept of something, that is repression of gene expression, takes place when you put double stranded RNA inside the cells. This initial idea came sometime in the year 1990. This was actually came from plant biology. Researchers working in the area of plant biology, they found that if you take plants like petunia, which produce beautiful colors, for example, here is a petunia flower, when they express the gene that actually codes for an enzyme involved in this production of this pigment, they actually expected that they should get a plant with flowers which are much more dark purple in color. So, this is a purple color flower. When you express a gene that is responsible for producing this particular purple pigment, when you overexpress this gene, you should actually get much more dark purple color flower. But instead, what they actually got is a flower which is paler, and there are actually many regions of this flower where there was no pigmentation at all. So, this was totally unexpected. So, instead of getting a flower with a dark purple, more dark purple color, you got a flower which is paler, and in fact, there is certain a variegated kind of a pattern in which certain areas of flower did not have color at all. So, this was very puzzling. So, how does introduction of additional copies of a gene involved in pigment synthesis result in decreased pigmentation? This was not expected. What you expected is this, but what actually happened was this. When they actually looked at the RNA, that is levels of RNA that is actually present for this particular gene in both the plant where they have introduced extra copies as well as the normal plant, surprisingly they found that if this is the level of mRNA for that particular pigment producing gene in the normal flower, when you introduce additional copies of this gene, instead of finding much more RNA, they actually found less RNA. So, this was against the dogma. You introduced extra copies of a gene and expressed it more, you should actually see much more RNA, but you ended up seeing less RNA, and as a result you got less pigmentation rather than more pigmentation. So, again the question is, how does introduction of additional copies of a gene lead to a decrease in messenger RNA levels? Because all in previous lectures we have been talking only about expressing genes. We want to make more protein, so we expressed how we can introduce genes into plants, how we introduce genes into yeast, insect cells, so that we can make much more of protein. But here now I am saying, here is a situation where I put extra copies of a gene, but instead of finding more RNA and more protein, I am finding less RNA and less protein. So, this is against what we have been discussing. So, let us now try to understand why this happened, why instead of getting a darker pigment and more messenger RNA, why you ended up getting less RNA and a paler pigment color. This phenomenon, which was first discovered in plants as well as in Neurospora, which is a fungus, at the time people did not understand what exactly is going on, because it beats the normal conundrum of the biology. So, they simply called it as post transcriptional gene silencing, PTGS. That is, after the RNA is formed, somehow the RNA is getting degraded. This phenomenon, which was originally found in plants like Petunia, was later observed in the fungus Neurospora causes, and it was called either quelling or PTGS, post transcriptional gene silencing. These things were happening sometime around 1990. For almost 8 years, people did not understand what exactly was going on, although people started describing this phenomenon in a number of physiological situations in variety of species. The answer actually came in the year 1998 by researchers working on a nematode called Sinorhabditis elegans. So, C. elegans is a very transparent nematode, it is a wonderful organism to study development biology. So, people are actually using this as a model system, model organism for understanding embryonic development, and when they were working with this, they unexpectedly stumbled upon a very interesting observation that actually opened up an entirely new area and led to the phenomenon of RNA interference. Now, what was this they observed? It was observed that when the double stranded RNA was injected into the worm, it led to a decrease in the expression of the homologous gene. This was the observation that Andrew Fire and Craig Mello, with the landmark paper was published in 1998, potent and specific genetic interference by double stranded RNA in Sinorhabditis elegans, and there was a news and views in Nature on this particular research article saying double stranded RNA poses puzzle, because this was nobody could explain this unusual phenomenon. Let us now see what exactly these authors did, what is the key experiment and the key result of this particular paper. This is what Fire and Mello reported in this paper. When they injected Sinorhabditis elegans, which is a nematode, with a double stranded RNA, and they observed a drastic decrease in the expression of a gene bearing homologous sequence. Now, injection of a single stranded RNA, either the sense strand or the antisense strand, had no or only marginal effect. These studies indicated that double stranded RNA, but not single stranded RNA, which people did not think normally existed in eukaryotic cells, had a biological function. This shook up the entire research community. So, I will just schematically show you what exactly is this experiment. This is the experiment. Here is the Sinorhabditis elegans worm. which is a transparent worm. You can just take a micro injection pipette and inject directly into cells of your interest, DNA, RNA or any protein or any solution. So, you can study a number of biological phenomena and study various regulators of gene expression. So, when they injected the sense RNA into this worm for a particular gene, the gene they have chosen is called as UNC22, and it turns out from earlier studies that if you reduce the expression of this UNC22 by other approaches, it resulted in a movement defect. So, as long as you have normal levels of UNC22, the worm was normal, but when you reduce the expression of this UNC22, the worm started twitching. It is called as a twitching behavior. So, it starts twitching. So, the levels of UNC2 determines whether the worm moves normally or whether the worm will twitch. So, what they did, when they took the sense RNA for this UNC22 and injected into the worm, nothing happened. When they took the antisense RNA, it had very little effect or a very marginal effect, or most practically can say no effect. But very interestingly, when you combine these two and injected a double stranded RNA, you started seeing a twitching effect. And they had evidence to show that when you injected this double strand RNA, it actually led to a decrease in the UNC22 messenger RNA and led to a decreased levels of UNC22 protein. And soon these observations were followed up with number of other genes in C. elegans. Here is for example another example, another mRNA called MEX3. Again in a normal animal, you can see this is the levels of MEX3, as is available, the dark blue color indicates the levels of the messenger RNA for this particular gene. Now, if you inject the double stranded RNA for this particular gene, you absolutely see no blue color, saying that there is no expression of the gene. So, what was initially observed with the UNC22 was later found to be true for a number of other genes, not only in C. elegans, but other variety of other systems like Drosophila, trypanosomes, mammalian cells and so on. Mammalian cells was not difficult, Drosophila and parasites and so on and so forth. So, this phenomenon of double stranded RNA induced gene silencing was termed as RNA interference or RNAi. So, when you inject double stranded RNA, which is homologous to a particular gene, into eukaryotic cells, it results in the loss of expression of that particular gene. This was the phenomenon. So, the double stranded RNA induced gene silencing or RNA interference or RNAi was soon reported for a wide range of eukaryotes ranging from worms, insects, mammals and plants. Here are some of the papers, like use of double stranded RNA mediated genetic interference to demonstrate that frizzled and frizzle2 acted in a wingless pathway, 1998. Targeted disruption of gene function Drosophila by RNA interference, a role for Nautilus in embryonic somatic muscle formation. Double stranded RNA induces mRNA degradation in trypanosoma brucei. Virus resistance and gene silencing in plants can be induced by simultaneous expression of sense and antisense RNA. So, this initial observation, the report by Andrew Craig and Mello, that Andrew Fire and Craig Mello, that when you introduce double stranded RNA into cells, it can result in shutting off of loss of expression of the genes, led to a flurry of papers, and people started demonstrating that yes, it is indeed true, not only for a particular gene in a particular organism, but in a variety of other organisms as well. Here are some more papers. Gene silencing in Neurophosphora cross-arc requires a protein homologous to RNA dependent RNA polymerase. I think I should come to this. So, people remember this phenomenon of overexpressing a gene leading to loss of expression was first studied in plants. Petunia, I gave an example of Petunia as an example. So, once Andrew Fire and Craig Mello reported that when you inject double stranded RNA, it leads to loss of expression of a gene, the plant biologists started relooking their initial observations, which they made in 1990s, and they soon discovered that the plants actually contain an enzyme called RNA dependent RNA polymerases that synthesizes double stranded RNA in presence of very high levels of RNA. This double stranded RNA in turn is responsible for the decrease in the expression of the homologous gene. So, the observation they made in 1990s of what happened in Petunia when you overexpressed these high levels of this pigment producing gene, instead of getting darker pigment you actually got a paler flower, now can be explained because when you put extra copies of a gene and when you produce large amounts of this RNA, instead of leading to higher amounts of protein, it actually led to the synthesis of a double stranded RNA, and this double stranded RNA in turn inhibited gene expression. That is why you had less of the protein. In fact, a flurry of papers came both in Neurospora as well as plants, reporting that this kind of a RNA polymerase capable of synthesizing double-stranded RNA is present. Like gene silencing, neuroprecursor requires a protein homologous to RNA dependent RNA polymerase. EGO1 is related to RNA directed RNA polymerase and functions in germline development of RNA interference and C elegans, Neurospora, C elegans, Arabidopsis. So SGS2 and SGS3 are required for post-transcriptional gene silencing and natural virus resistance. Again in Arabidopsis, an RNA dependent RNA polymerase gene is required for post-transcriptional gene silencing mediated by a transgene but not by a virus, and on the role of RNA amplification in double-stranded RNA triggering gene silencing. So you can see a variety of organisms soon found to have this RNA dependent RNA polymerase, which is capable of synthesizing double-stranded RNA when large amounts of RNA is synthesized in the cells of these organisms. So this observation that when you overexpress, when you make too much of RNA of a particular gene, that can actually have a negative effect on gene expression. It can actually result in repression of gene expression, was soon known as knockdown. So the repression of the decrease in the expression of a gene as a result of introduction of double-stranded RNA or as a result of RNA interference is known as knockdown, as opposed to knockout, which we studied in the previous class. Knockout is primarily because of loss of a gene or a deletion of a particular gene. That is what we studied in the last class. Whereas knockdown, the gene is very much there, but due to a post-transcriptional mechanism, there is a decrease in the levels of RNA leading to decreased level of protein. So this is called as knockdown versus gene deletion leading to decreased expression, which is called as knockout. So RNA interference results in knockdown, not knockout. Now, lot of effort then went on to understand how does introduction of double-stranded RNA into cells can result in decrease in gene expression. To cut a long story short, enormous amount of effort was spent in understanding the mechanism. I am just trying to simplify the whole thing and giving you a very schematic representation of what was the final outcome and what we understand at present, the mechanism by which when you introduce double-stranded RNA inside the cells, how does it result in the decrease in gene expression and less amount of a protein. Now what happens? First, the double-stranded RNA teams up with a protein complex called as dicer, which cuts the long RNA into short pieces. So this is the first thing that happens. So whenever large amounts of double-stranded DNA or RNA is introduced inside the cell, this double-stranded RNA is recognized by a multi-protein complex called as dicer. Remember, it is not a single protein, it is a multi-protein complex known as dicer. This dicer recognizes the double-stranded RNA and cuts this double-stranded RNA so that you get smaller RNAs. Then another protein complex called as RISC, which is an acronym for RNA induced silencing complex. RISC is RNA induced silencing complex. This RISC now takes up this double-stranded RNA, discards one of the two strands and takes up only one. This is what happens. So the dicer chews the double-stranded RNA into small pieces and then RISC takes one of these RNAs. Of the two, it only takes one of the RNAs, and this RNA then, the RISC docked single-strand RNA then pairs with the messenger RNA which is present inside the cell by homologous base pairing, and once the RISC in the RISC complex, when this RNA, small RNA pairs with mRNA, that results in the cleavage of the messenger RNA leading to RNA degradation and gene silencing. So this is finally the mechanism that was discovered, the mechanism by which introduction of double-stranded RNA inside the cells leads to mRNA degradation and therefore results in the loss of protein or decrease in the levels of the protein. The same thing I have shown it in a very schematic manner here. So when you have double-stranded RNA inside the cells, this double-stranded RNA is recognized by this dicer complex, which then cuts them into small RNAs called small interfering RNAs. These small RNAs produced by dicer are known as siRNAs or small interfering RNAs, and then one of the two strands of this siRNA is now loaded on the RISC complex, RNA induced silencing complex, and this, when it hybridizes the messenger RNA which is present inside the cell, the messenger RNA is cleaved and therefore the messenger RNA is degraded and therefore the protein cannot be produced. So this is the mechanism by which gene silencing is brought about by RNA interference pathway. Now, as I said, the dicer is a multi-protein complex. A lot of effort has gone into understanding the key components of this dicer complex, and one of the key components of this dicer is called as argonate, which is a central component of this RISC or RNA induced silencing complex, and one strand of the double stranded RNA that is produced by the dicer. Remember, the double stranded RNA gets chopped by dicer into smaller pieces, and one of the strands that is produced by double strand is retained in the RISC complex in association with argonate, and the other strand is what that is loaded on the RISC complex. Therefore, it can pair with messenger RNA and then degrade the messenger RNA. So, induction of RNAi using double stranded RNA quickly became a very powerful tool for the study of function of genes in many lower eukaryotes, especially C. elegans and Drosophila melanogaster. So, you can see it became very powerful tool, because if we want to understand a particular function of a particular gene, all that you have to do is make double stranded RNA for that gene and inject into this worm, or you can even just make a plasmid construct expressing double stranded RNA and take this bacteria and simply feed this worm actually feeds on bacteria. So, if the worm just eats this bacteria which are making this RNA, then also gene can silenced. So, you can either inject the double stranded RNA into these worms, or you can just feed them. That results in loss of expression of the particular gene. So, instead of doing knockouts, making the knockout constructs and then doing all kinds of gimmick to introduce the genes and then knock down the gene expression, here was a very simple method by which you can knock down the expression of a particular gene by simply injecting double stranded RNA into these organisms. However, this kind of a knockdown using RNAi was not possible in mammalian cells, because there was a huge problem. In mammalian cells, when you introduce this double stranded RNA, the double stranded RNA actually introduces what is called as a immune response leading to apoptosis. Now, this is a kind of a immune mechanisms that eukaryotes have evolved, because whenever the eukaryotic cells are infected by viruses, these viruses as part of their life cycle actually produce double stranded RNA, and whenever double stranded RNA is made inside the cells, this double stranded RNA activates what is called as interferon pathway, leading to apoptosis, so that these virus infected cells are destroyed and thereby preventing the spread of the virus to neighboring cells. So, this is a kind of a protective mechanism that eukaryotic cells have evolved to recognize virus infected cells, because it is only the virus infected cells that is double stranded RNA is made, and therefore, whenever double stranded RNA is made inside, the cell recognizes that a virus has infected and therefore immediately activates this dicer mediated mechanism and cleaves the viral RNA, double stranded RNA, so that the virus cannot multiply, and therefore spread of the virus infection to remaining cells is abolished. So, because of this problem, this kind of a double stranded RNA injection or introduction of double stranded in mammalian cells was not possible to do RNA interference in mammalian cells. So, the mechanism of destruction of double stranded RNA is used by mammalian cells during viral infection by destroying virus infected cells and preventing the spread of the virus to neighboring cells. Thus, for quite some time, RNAi or RNA interference using double stranded RNA was largely confined only to lower eukaryotes. So, you could study gene functions, or you can do RNA interference only in lower eukaryotes like insects and worms, but you could not do this double stranded RNA induced gene silencing in higher eukaryotes. Then another interesting observation came. Then a new strategy was developed by Schull and his co-workers. What they actually showed is that you can trigger RNA interference in mammalian cells by using small double stranded RNAs instead of long double stranded RNAs. So, what the Schull and his co-workers actually demonstrated is that instead of using long double stranded RNAs, if you use RNAs of about 20 to 22 base pairs, then you can trigger RNA interference in mammalian cells, and these small RNAs do not induce the antiviral or the immune mechanism leading to destruction of the virus of the cells and does not trigger apoptosis. So, the key for the use of RNA interference in mammalian cells came from the observation that do not use long double stranded RNAs, but use short double stranded RNAs which are about 20 to 25 base pair cells, and now you can trigger RNA interference in the higher eukaryotes or mammalian cells also. So, these small double stranded RNAs were ultimately known as small interfering RNAs or siRNAs. So, this is the origin of the siRNAs. So, the discovery that large double stranded RNAs induce apoptosis or activate the double strand mediated degradation of the RNAs and results in destruction of the cells, and therefore, RNA interference cannot be done in mammalian cells, but if you use small RNAs or what are known as the small interferon RNAs, they can actually bind to messenger RNAs and silence the homologous gene. So, this was the origin of this terminology siRNA or small interfering RNAs. This actually opened the door for knockdown of genes in mammalian cells, leading to exploitation of RNAi both for basic research as well as a therapeutic tool. So, let us now try to understand some of these important experiments done in the eukaryotes. So, Synthetic siRNAs are typically 21 base pair double stranded RNA molecules with dinucleotide 3' overhangs. When you introduce these short 21 base pair double stranded RNA with 2 base pair overhangs at the 3' ends, these synthetic RNAs are directly loaded onto the RISC complex, and upon introduction to the cytoplasm by transfection or electroporation, and then it leads to RNAi. So, you can degrade specific messenger RNAs, or you can silence specific genes in higher eukaryotic cells by introducing small double stranded RNAs known as siRNAs, which are about 21 base pairs in length with 2 base pair 3' overhangs. And when you introduce these short double stranded RNAs or siRNAs into mammalian cells by either electroporation or by using transfection techniques, these RNAs are directly loaded out of the RISC complex, and one strand is then base paired with messenger RNA, leading to degradation of that particular messenger RNA. Therefore, instead of using long RNAs, if we use short RNAs, RNA interference can be demonstrated in mammalian cells as well. So, if you want to now do RNA interference, if you want to knock down the expression of a particular gene in mammalian cells by RNA interference, you can do it in 3 different ways. Number 1, in non-mammalian systems, the RNAi pathway is turned on when double stranded RNA is introduced into the cells. This has been very useful, especially people working in the area of insects such as silkworms and Drosophila, because you can simply knock down the expression of a particular gene in these insects by simply feeding or by simply injecting the double stranded RNA into these lower eukaryotes like worms and insects. So, you can simply, here size does not matter, you can inject as well as the long double stranded RNAs, because it does not activate the immune system there. It can directly be digested by the dicer into smaller fragments and then loaded on the RISC complex, leading to silencing of the particular gene. This is one method. However, if you want to do the RNA interference in mammalian systems, there are 2 other methods by which you can do the RNA interference. In one case, in mammalian systems, RNAi can be triggered by introducing DNA based expression vectors designed to express short hairpin RNA or shRNA molecules, or by direct transfection of synthetic short siRNA molecules. So, by introducing plasmid that actually can make what is called as shRNA, which is then cleaved into generating siRNA, or by directly expressing siRNAs, you can actually induce RNAi or RNA interference in mammalian cells. Either you express these plasmids expressing this what is called as a short hairpin RNA, which are then converted to siRNAs inside the mammalian cells, or you can directly inject siRNA molecules. In both these cases, gene silencing results from the destruction of the messenger RNA that is complementary to the input siRNA, or the siRNA molecules created by the dicer, cleavage of long RNA double stranded RNAs and shRNA molecules. So, this is the mechanism by which you can do RNA interference in mammalian or non-mammalian cells. Now, so this triggered a whole bunch of experiments, because people want to know, especially in the era of human and mouse genomes being sequenced, people want to understand the function of a number of genes, and the best method of understanding the function of many of these genes is to knock down the expression of these genes using siRNAs. So, if you want to understand, for example, what is the function of a particular protein in a particular signal transduction pathway, all that you have to do is to make a short 21 base pair siRNA for that particular gene and transfer them into cell lines, knock down the expression of them, and see what happens to the particular signal transduction pathway. So, this is just one example. So, but the problem came, we soon realized that when you introduce these short 21 base pair RNAs, siRNAs into the cells, many times we had what is called as off target effects. That means, in addition to specifically inhibiting that particular gene which is complementary to the siRNA, the siRNA also had non-specific effects, and it non-specifically repressed the effects expression of a number of other genes as well. So, these are known as off target effects of RNAi. So, it was observed that RNAi could also change the expression of RNAs that closely resemble the target RNA and not just the target RNA itself. So, the excitement for a while died down. So, soon it became very clear that the RNAi can change levels of protein that are not related to the target RNA. So, the euphoria that was there initially saying that siRNA can be used for knocking out the expression of specific genes kind of died out when people realized that the effect that you are getting, the ultimate effect you are getting, for example, if you take do a micro array experiment. If you take the genes which are expressed in a normal cell and genes which are getting expressed in a siRNA transfected cell and see what are the changes suppose a number of genes are getting either upregulated or downregulated, it is not because your siRNA specifically knocked down a particular gene, but it also hybridized a number of other genes, and therefore the effect is non-specific and not actually specific for that particular target gene. Now, how do you overcome this? There is a very nice review on trends in genetics, off target effects of RNAi, that actually talks extensively about this off target effects of RNAi. So, how to avoid this off target effect of RNAi? That means you want your siRNA to knock down the expression of only your gene of interest, but not other related genes. How do you do that? So, people started developing algorithms. So, if you want to now make a 22 base pair siRNA for a particular gene, you make sure this 21 base pair RNA does not hybridize to any other gene in the mouse genome or human genome. So, powerful algorithms were developed to make sure that the siRNA, the sequence that you have chosen, is highly specific for only your target gene, and it does not go and hybridize related genes and therefore knock down. So, you have to score for those regions of the gene which do not share any homology with the rest of the sequence in the human genome. Therefore, when this set of segment of RNA hybridizes, it can only hybridize to your gene of interest, and it does not provide any off target effects. So, careful design of siRNA is required to maximize silencing of the target gene and minimize the off target effects. With improved understanding of the mechanism of RNAi, algorithms for designing effective siRNAs were developed, decreasing the number of sequences that need to be tested for eliciting gene silencing. So, it became clear, for example, if you want to show that if I knock down the expression of this particular gene by using siRNA, and if I am getting this particular phenotypic effect, people said you cannot just show this effect with one siRNA, you at least design siRNA for 3 or 4 different regions of this gene and show that you get a same effect. Then only we believe the result. So, just by developing one siRNA for a long gene and showing that it results in a particular phenotypic effect was not acceptable. So, you have to design at least 2 or 3 different siRNAs encompassing different regions of the gene, and if they all have the same effect, then you start believing that yes, this effect is actually very specific and it is likely the effect due to the downregulation of that particular target gene. So, it was necessary to independently test multiple distinct siRNA sequences targeting the same transcript to confirm that the observed gene silencing is due to specific rather than off target effects. Today, there are actually companies which are making siRNAs for almost all the genes of mouse or humans or other mammals. You can just buy siRNA. Suppose I want to knock down the expression of a particular gene, I can just go to a company like QIAGEN or Dharmacon, and there are so many companies now. You can just go then order siRNAs for that particular gene. They will simply ship you 3 or 4 best siRNAs, and they guarantee you that if you introduce these siRNA into the cells, you get more than 80% knockdown of that particular gene. Otherwise, they will give you the money back. So, a lot of algorithms were developed. Lot of work has gone on to make sure that the siRNA that are designed will not have any off target effects, and they exactly knock down your target gene, and therefore, you will get very significant knockdown of your gene of your interest. Now, so far we discussed about introduction of exogenous double stranded RNA, that is, when you introduce exogenous double stranded RNA, how it gets broken down to small 21 base pair RNAs, and then how it hybridizes to messenger RNA by the RISC and then results in the knockdown of the gene expression. But then the question came, do such small RNAs, are they naturally present in the eukaryotic cells? Are they naturally made? So, double stranded RNAs were actually found to be naturally produced in eukaryotic cells during development, and they were found to have key role in regulating gene expression by acting as negative regulators of gene expression. So, you can see initially the entire RNA interference was studied using RNAs which were introduced into the cell from an exogenous source. You either introduce a double stranded RNA into the cell, or you either introduce a small double stranded RNA into the cell, and that inhibited the target gene expression. But soon it became clear inside naturally many such small double stranded RNAs are actually produced, and these double stranded RNAs that are produced naturally can have very important regulatory functions. So, these double stranded RNAs which are processed by dicer machinery to generate small single stranded RNAs are known as micro RNAs or miRNAs. So, I am introducing a new terminology here. So far we discussed about siRNAs, which are small interfering RNAs, which are actually generated by the cleavage of exogenously introduced double stranded RNA. But here I am telling you that small double-stranded RNAs are also generated endogenously, and these endogenously generated small RNAs which are capable of inhibiting gene expression are known as miRNAs or microRNAs. So, I have siRNAs and I have miRNAs. The first naturally occurring miRNA or microRNA was again discovered in the worm C. elegans, and this miRNA was known as Lin4, which is about 22 base pairs long. This was actually identified in a screen for defects in the timing of larval development, and Lin4 was partially complementary to the conserved sites in a very important regulatory gene known as Lin14, 3' untranslated region of this Lin14 RNA. Therefore, when this Lin4 microRNA goes and hybridizes to the Lin14 untranslated region, it results in the knockdown leading to the reduced expression of that particular gene. Lin4 actually binds to the sites and negatively regulates Lin4 translation. So, translation is inhibited and therefore, protein is not made. So, the first naturally occurring microRNA Lin4 was actually discovered in the year 1993, and for almost 7 years no other microRNA was discovered. It took almost 7 years to discover the second microRNA, again in C. elegans. This is about 21 bases long. This was named Let7 and was discovered by Reinhardt et al. In the year 2000, which also regulates Lin14 RNA in the same way as Lin4. So, the small RNAs of 21 20 base pairs are also occurring naturally, and they also seem to have a very important role in the regulation of embryonic development in C. elegans. Soon, microRNA genes were discovered in a number of other organisms, including Drosophila, mammals, plants, and so on and so forth. And these microRNA, there are actually genes which actually code for microRNAs, and these microRNA genes are actually transcribed by RNA polymerase II as long double-stranded RNA precursor called primary microRNA or pri-microRNA. So, just like you have protein coding genes, that is, you have genes which code for messenger RNA which then code for proteins, there are actually genes in the eukaryotic genomes which actually synthesize precursors for microRNAs. These precursors are called primary microRNA or pri-microRNA, which are a single RNA which folds into number of hairpin structures leading to a long precursor of microRNA. The microRNA portion of this primary microRNA transcript forms a hairpin which signals for double-stranded RNA specific nuclease cleavage. So, when you have these long RNAs with number of stem loop structures, these specific hairpin structures are actually recognized by a specific double-stranded RNA cleaving enzyme. Actually, it is an RNase 3 type of enzyme. This enzyme is known as DROSHA. A double-stranded RNA specific ribonuclease known as DROSHA actually processes this primary microRNA to release this hairpin into a precursor microRNA or pre-microRNA. So, you have a gene, microRNA gene, from that you get the pri-microRNA or a primary microRNA, and in this primary microRNA, the microRNA regions are cleaved by DROSHA to generate what is called as a precursor microRNA or pre-microRNAs. These pre-microRNA then exported into the cytoplasm by a nuclear export protein known as exportin 5. And once in the cytoplasm, this pre-microRNA is cleaved by the enzyme dicer, which we discussed earlier, into a 20 to 25 base pair long double-stranded RNA. So, the hairpin structures are removed and you now get a 22 base pair, a 20 to 25 base pair double-stranded RNA. These double-stranded RNA produced by dicer are then separated. The single-stranded mature microRNA assembles into a protein RNA complex, namely the RISC complex, which we discussed earlier. And this RISC complex, through the RISC complex, the microRNA targets the messenger RNAs by direct base pairing, leading to knockdown of the expression. The 5' region of a microRNA, especially nucleotides 1 to 8, is the most critical for targeting and function. The microRNA target sites are often imperfect matches and located in the 3' untranslated region of target messenger RNAs, and since the microRNAs do not require perfect complementarity for target recognition, a single microRNA is able to regulate multiple messenger RNAs and often produce measurable phenotypes. So, unlike the siRNA, which are highly sequence specific because we are actually introducing to the cells, many times this microRNA need not have an exact complementarity, it can even go and hybridize with 1 or 2 mismatched bases, and therefore, it can have a very drastic phenotype, a measurable phenotype. So, a single microRNA can go and inhibit the expression of a number of RNA molecules. So, whatever I showed here in the form of a text, I have now shown it actually in the form of a cartoon here. So, there are actually genes inside the eukaryotic cells which actually code for, which are actually responsible for generation of microRNAs naturally in eukaryotic cells. These genes are actually transcribed by RNA polymerase II, and you get what is called as a microRNAs or primary microRNAs, and these primary microRNAs are recognized by an enzyme called as DROSHA, which is an RNase, and it cleaves to generate what is called as a precursor microRNAs, and these precursor microRNAs are then exported into the cytoplasm by a protein called exportin 5, and once the precursor microRNAs are in the cytoplasm, it is recognized by the dicer complex, and the dicer complex now cleaves the hairpin, generating the double stranded RNA, and one of the strands is then hybridized to the mRNA in the RISC complex, leading to degradation of the specific messenger RNA. So, the mechanism of miRNA and siRNA action more or less similar, whereas siRNAs are actually generated from an exogenous source, whereas miRNAs are generated naturally inside the cells. MicroRNAs have been shown to play integral role in numerous biological processes, including immune response, cell cycle control, metabolism, viral replication, stem cell differentiation, and human development. You name a physiological processes, microRNA seems to be playing an important role in all these processes. Many microRNAs are conserved across multiple species, indicating the evolutionary importance of these molecules as modulators of critical gene functions. MicroRNA expression or function was shown to be significantly altered in many disease states, including cancer, heart failure, and viral infections. So, it soon became clear that these microRNAs, which nobody heard of these microRNAs in the early 1990s, but once people realized that these small microRNAs are actually made naturally inside the cells, and they have important regulatory functions, it opened up an entirely new area of research in biology and medicine. So, I will just summarize some of the key points I have discussed so far, and then we will proceed further. What we have discussed so far is that small RNAs in the form of siRNAs and miRNAs play large roles in the regulation of gene expression in eukaryotes. They are very important for normal cell metabolism, development, and defense against invaders such as viruses. The siRNAs or small interfering RNAs are produced from longer segments of double stranded RNA by dicer, assembled into RNA-induced silencing complex or RISC, and targeted to messenger RNAs with perfect complementarity, giving silencing by cleavage and degradation of the RNA or by formation of a heterochromatin. We will discuss about this heterochromatin later, it is a little bit more complicated. The pathway for microRNA has many steps in common with that of siRNA, because both are generated by the dicer mechanism and RISC mechanism, but miRNA is produced from an endogenous source, siRNA is produced from an exogenous source. miRNAs are processed from hairpin structures by DROSHA and then by dicer, and they most often have imperfect complementarity with their targets. This is another important difference between siRNAs often have a perfect complementarity with their targets, miRNAs often have imperfect complementarity with their target sequences. The miRNA is a single strand RNA of about 22 nucleotides in length. It is generated by the RNase 3 type enzyme DROSHA and dicer from an endogenous transcript that contains a local hairpin structure. It originates as a primary RNA of several hundred base pairs. So, you have a long primary RNA from which these hairpin structures are cleaved. A single primary RNA can actually give rise to a number of precursor microRNAs. Okay. So, a single microRNA gene, many microRNAs can arise from a single primary microRNA gene. MicroRNAs again contribute significantly to the regulation of cellular gene expression. Silencing of the endogenous genes regulates basic biological processes, including transition from one development stages to next. Many of these microRNAs are actually expressed in a developmental stage specific manner. Some of the microRNAs which are originally discovered, like the LEN7 and LIN4, regulate C. elegans larval development, and miRNAs are expressed in a specific spatial and temporal pattern during development in Drosophila melanogaster and differentiation of mouse embryonic stem cells. And in fact, many of the microRNAs, their function still remains unknown. Say, an entirely new area of research opened up with the discovery of this RNA interference mechanism, and that is why it became a very important discovery, and Andrew Fire and Craig Miller got the Nobel Prize for this work. So, what are the major differences between miRNA and siRNA? miRNAs are nothing but microRNAs. They are encoded by endogenous genes. They are hairpin precursors. Their hairpin precursors are called pre-microRNAs. These pre-microRNAs with imperfect complementarity in their stems and frequent bulges, as well as mismatches and wobbly base pairs, they can ultimately recognize multiple targets. Whereas siRNA is short interfering RNA or small interfering RNAs. They are mostly exogenous origin. They are double stranded precursors. They may be target specific. siRNAs are often target specific, miRNAs may have multiple targets. So, siRNA and miRNA are discovered in different ways. One is discovered through an exogenous source, another was an endogenous source, but their biogenesis is more or less similar. Both involve dicer and RISC complexes for their function, and they share common pathway components and they have common outcomes. Both of them inhibit degrade RNAs and inhibit gene expression. So, you can see this discovery of RNA interference opened up an entirely new area of research, and no doubt this RNA interference was recognized in a very important biological process for modulating eukaryotic gene expression, and is actually named as breakthrough of the year in 2002 by this prestigious science magazine. So, small RNAs became very important for regulation of gene expression, as well as for understanding gene function, as well as for potential therapeutic agents for a number of diseases. So, from being named as Science Breakthrough of the Year in 2002, advances in our understanding of the natural role of RNAi in a range of cellular processes, both protective and destructive, have revealed the potential for a novel class of drugs based on small inhibitory RNAs and derivatives. We will discuss some of the potential applications of RNAi as we go along. The natural ability of eukaryotic cells to repress gene expression via RNAi was exploited for silencing target genes in vitro, in tissues, as well as in whole organisms. So, this area opened up an entirely whole range of research areas for understanding gene functions as well as number of cellular processes. So, let us now spend some time to understand what are the benefits of this RNA interference. So far we discussed how RNAi was discovered, how siRNAs were discovered, how miRNAs were discovered, what are the differences between the two, and how the discovery of these siRNAs, miRNAs changed the entire biological research and became very important tools for inhibiting gene expression and for understanding the roles of a number of genes. Now, let us see what are the benefits of RNAi. Now, people started talking about what is called as RNAi therapy. Now, in our earlier lectures we had discussed what is called as gene therapy. Now, gene therapy was restricted to diseases such as hemophilia or cystic fibrosis, where a normal copy of a gene could replace a mutant gene and cure the disease. This is what we discovered in our lectures on gene therapy, where if there is a mutant copy of a gene, if a normal functional protein is not made, you introduce the gene into the humans and see whether it can make the normal protein and cure a disease. But the discovery of RNA interference actually opened up the possible gene therapy for diseases caused by overproduction of a normal protein. Now, in the gene therapy we actually discussed about a missing protein or a mutant protein. So, in such cases if you express a normal protein, you cure the disease. But there are also many disorders where overexpression of a normal protein can lead to a disorder. Many neurodegenerative disorders are actually caused by overexpression of a mutant protein. So, if you can somehow block the overexpression of this mutant protein, you can cure a disease, and RNA interference became an excellent tool for doing these kinds of, or for preventing the repression of the expression of these mutant proteins. So, RNAi could be harnessed to block viral infections, to stop the overproduction of a protein that drives diseases like macular degeneration, which is a leading cause of blindness. We will discuss some of these things in detail. I am going to give some 4 or 5 examples now, just to tell you what are the applications of RNAi. Here is a paper that appeared in 2004 in Nature Medicine, talks about RNAi suppresses polyglutamine induced neurodegeneration in a model of cyanocerebellar ataxia, which is a very important neurodegenerative disorder. Now, SCA1 or the spinocerebellar ataxia type 1 as well as other neurodegenerative disease called Huntington disease, they are progressive untreatable neurodegenerative disorders caused by repeat of polyglutamine repeats. I will not go to the details, but what is called as a polyglutamine repeat expansion that actually leads to this neurodegenerative disease. So, people ask the question, can we prevent expression of these polyglutamine containing proteins by RNA interference? In fact, in inducible mouse models of SCA1 and Huntington disease, repression of the mutant allele expression improves disease phenotypes. Thus, therapies designed to inhibit the expression of the mutant gene will be beneficial. So, people found out if we can repress or inhibit the expression of these mutant proteins which have huge polyglutamine repeats, you can actually cure the disease. So, what people have done is, upon intracerebellar injection, recombinant adeno-associated viruses vectors expressing the shRNAs, which can ultimately lead to the generation of siRNAs, was found to improve motor coordination, restore cerebral morphology, and resolve the characteristic ataxin inclusions in Purkinje cells of SCA1 mice. So, by inhibiting the accumulation of these mutant proteins, by injecting siRNAs complementary to this gene coding for the polyglutamine repeat containing proteins, you can actually demonstrate clinically beneficial results, clinically significant results, indicating that siRNAs can be used for potential therapeutic applications. So, these data demonstrate that RNAi can be used as a potential therapeutic tool for number of neurodegenerative diseases. Here is another paper that said lentiviral mediated silencing of SOD1 through RNA interference retards disease onset and progression in a mouse model of ALS. Now, mutations in the superoxide dismutase coded by the SOD1 gene is one of the major causes of familial amyotrophic lateral sclerosis, which is a very complicated neurodegenerative disorder leading to progressive death of motor neurons through gain of function mechanism. And in this paper they show in a SOD1 transgenic mice, a model for the familial ALS, intraspinal injection of a lentiviral vector producing RNA mediated silencing of SOD1 results in a decrease in SOD1 protein, leading to retardation of both onset and progress of the disease. There are again other examples here: inhibition of respiratory viruses by nasally administered siRNA. In one case they express siRNA by adeno-associated virus, in another case they express siRNA by lentivirus. Here you can simply nasally administer siRNA and show it has a therapeutic benefit against certain viral infections. Here another very interesting paper, a company called Anilam Pharmaceuticals actually can show RNA can actually lower cholesterol levels in mice. You can go to the website and then read more about this. There is also a nice article on Nature about how you can actually bring down cholesterol levels by RNAi. Another company called Acuity Pharmaceuticals is trying to develop RNA based therapeutics, where this company hopes to use small interfering RNA to treat patients with wet age related macular degeneration, a condition that is caused by extended growth of blood vessels inside the retina, by actually inhibiting a protein called as VEGF, vascular endothelial growth factor. So, RNA interference had a tremendous applications in both basic and applied research. For example, you can use RNAi for testing the hypothesis of a gene function. RNA interference allows scientists to knock down expression of a targeted gene and observe the effects of what happens when partial or fully lock out the function of a particular gene. I will just give an example here, it is appeared in JBC in 2005. For example, interleukin 1 receptor and the Toll-like receptor induce mitogen activated protein kinase pathways and activate transcription factors such as NF-κB and AP1. Now, 3 members of a protein family called pelino protein family were shown to be mediators of these pathways, but show specific effects in these pathways. Now, you want to understand what happens if I knock down one of the members of this pelino protein. How does it affect the signal transduction pathway? So, when you use siRNAs targeting human pelino 3, there are 3 pelinos target only pelino 3, it was shown to have a specific physiological role in mediating IL-1 activation of P38 MAP kinase in these cells. So, by knocking down specific components of a signal transduction pathway, you can exactly understand what is the function of that particular component in the entire signal transduction pathway. You can use a RNAi for functional screening and target identification. For example, cellular pathways have been characterized biochemically or genetically can be further dissected using RNA interference. Libraries of siRNAs targeting large collections of genes enable screening experiments that tie genes to cellular function. I will give you one example. For example, a collection of siRNAs targeting about 650 known putative kinases was screened in HeLa cells using a histone DNA fragmentation ELISA to provide a quantitative cell based assay for apoptosis. That is, what are the genes that are involved in what are the kinases that trigger apoptosis. So, you make siRNAs for each one of these kinases, transfer them into cell lines and see which of them block apoptosis. And they identified 73 kinases, that is 11% of this kinase library, and when you knock down these kinases using siRNA, there was an increased apoptosis, and therefore these kinases were termed as survival kinases. So, you can see by using siRNA you can actually identify those kinases which can actually trigger apoptosis. Which can increase apoptosis. Target validation. The normal drug development involves target identification, validation, assay development, drug lead identification, lead optimization and testing and so on and so forth. Now, once a potential therapeutic target has been identified through a variety of biochemical genetical methods, screening can be done by using RNAi, and siRNAs can be used to reduce its expression and see whether this can really be a valid target. Just now I showed you, we can actually use by injecting siRNAs against a protein called as vascular endothelial growth factor into the eye, you can actually prevent a disease called macular degeneration, which is a major cause of blindness especially in aged people. So, by inhibiting expression of the VEGF in the eye, you can actually improve vision and prevent this decrease the effect of this macular degeneration. I am not going to the details of this paper, very nicely discussed, and the paper appeared in Gene Therapy, suppression of ocular neovascularization with siRNA targeting VEGF receptor. siRNA as a therapeutics. A number of RNA based drugs are currently in preclinical development for targeting respiratory syncytial virus, hepatitis C, HIV, Huntington disease, and many neurodegenerative disorders, some of which we have just discussed. This is what I just discussed here. For example, in this case, 84 siRNAs targeting mouse and human apolipoprotein protein were screened, and 2 of them actually were found to decrease the levels of apolipoprotein B messenger RNA, and this apolipoprotein B actually binds the LDL receptor and therefore essential for the formation of low density lipoproteins. And if you decrease the apolipoprotein B mRNA, you can prevent LDL formation, and therefore it can have a therapeutic effect and can prevent atherosclerosis. A very nice paper, therapeutic silencing of an endogenous gene by systemic administration of modified siRNAs. By simply injecting these siRNAs against apolipoprotein, you can reduce your cholesterol levels, LDL levels. The SOD I again discussed, I will not go into the details, one can actually read. Again, a very nice paper, lentiviral mediated silencing of SOD1 through RNA interference retards disease onset and progression in a mouse model of ALS. Again, how viral infections can be inhibited by using siRNA as therapeutics. A number of new small RNAs are being discovered, and many novel functions are being discovered for RNAs. For example, again due to lack of time I will not go to the details. siRNAs again has been shown to be involved in the formation of heterochromatin. A new siRNA called as repeat associated RNA or RNA has been shown to actually activate heterochromatin formation in eukaryotic cells. That in turn can induce gene silencing. Many small RNAs called tiny non-coding RNAs or TNC RNAs, whose function is still not clear, have been identified. Small modulated RNAs, mRNAs, which actually turns a transcription repressor into activators, are being described. So, number of applications have come out. So, I think I will stop here, and I have just listed in the next few slides some of the key papers. The Nobel lecture by Andrew Fire is an excellent source for understanding more. This is the first paper that I described the double strand induced RNA gene silencing, and there are a number of key developments that took place in the area of RNA interference, which I have listed here. Some of the very nice review articles in Nature, collections on microRNAs. One can just click onto the website and then read some of these articles, and I have listed some more very interesting articles in the area of RNA interference. And there is also a very nice movie on RNA interference. One can just click on the Nature animations, and you can actually visualize how exactly RNA interference takes place by watching this nice movie. I think I will stop here. Thank you.